we are going to welcome Michael Bronstein. He's going to discuss the topic on harnessing geometric machine learning for molecular modeling. And he's a Deepmind professor of AI at Oxford University. So, though he can hear you, but you will not be able to hear the applause, I request you a pretty energetic round of applause, please, to begin the day. Welcome, Michael.
Thank you very much. I hope you can hear me. That's also my first time in such a room. And I would like to talk today about the use of machine learning and artificial intelligence in biological fields. And well, you're probably all excited as I am from the last week in Stockholm and the Nobel Prizes that were awarded that propel very strongly this field. So when we talk about biology, if we look at our own body, basically, we can look at ourselves at very different scales, from the molecules, like small molecules, proteins, that build up then cells, tissues, organs, organisms, and populations. So there is a huge gap of multiple orders of magnitude, both in spatial dimensions and in temporal dimensions, across different levels of abstraction that we can think of biology. And if we take a typical cell, let's say when a drug acts on a cell, it consists of only maybe 10 atoms, maybe 20 atoms. A typical protein molecule might have already tens of thousands of atoms, and the entire cell might have something like quadrillion of atoms. So it's huge, again, huge gap between the different orders of magnitude. And the methods that we can apply to model, for example, how a drug works, already will not scale to proteins. So we'll need to use a different level of abstraction, different mathematical and physical principles, and also we cannot scale to the entire cell, let alone the entire body. The issue is that at the very lowest level, we understand very well how these systems work, right? So we can solve physical equations like the Schrödinger equation to model drugs. We cannot apply them, it's computationally intractable, we cannot apply them to different molecules. And also, this was a very famous work by Philip Anderson, where he says that you cannot start with fundamental laws that govern the basic levels and try to apply them to higher levels. Instead, there are different laws that emerge at different scales. The problem that we typically don't have a good understanding or a good description of the laws at the higher levels. And this is really a big problem in, well, not only in biology and computational sciences in general, because when we want to computationally solve some problem, let's say in biology or in biochemistry, our basic premise, our assumption is that we know the laws that govern this problem, or at least we have the best understanding of this model. We then program these laws in the form of mathematical equations and then logical instructions for a computer, and then we can apply it to new data and basically produce some output that at least approximately describes the behavior of the system. Now, when we try to model at high level of abstractions, if we don't know how to describe the laws, right, we don't have an equation of a cell, for example, then this approach cannot work. And that's why AI systems, they actually operate in a sense that they reverse this order. So the AI system is shown examples of behavior of a system, the inputs and outputs. And then it learns with appropriate algorithms, with appropriate compute, to fit some kind of black box to these data. So in this sense, the instructions to the computer are not explicitly programmed. They programmed implicitly, usually through the weights of a neural network that implements this system. And this has really been this departure from handcrafted instructions to something that is data-driven and learned was really the big success of deep learning methods in the last decade. And in a nutshell, if you think of simple setting of AI or machine learning, right? So like image classification, basically you can think of it as a kind of glorified function fitting. So I give you examples of cats and dogs, right? Because it's very difficult to mathematically describe what makes a cat look like a cat or a dog look like a dog, but I can give you many examples of such cats, right? And basically what we try to do is we try to find a parametric family of functions that represents, well, that allows to distinguish cats from the dogs, and we automatically learn the features that characterize these classes of images. Now, we already know from the 50s that neural networks can implement a very broad family of such functions. We know that they have what is called universal approximation property, so more or less any function can be represented by such systems. But the computational problem we run into is what is called the curse of dimensionality. So in practice, the number of samples or a number of training examples that we need to show to such a system grows exponentially with the dimension. And in most machine learning problems, we deal with dimensions that are very high, hundreds or thousands or maybe millions. So the number of cats and dogs can be more than the number of particles in the universe. So in computer vision and pattern recognition, the remedy or the workaround this problem of curse of dimensionality actually was inspired by works in neuroscience, the Nobel-winning work of Hubel and Wiesel, who studied the organization of the visual cortex of mammals and inspired a new class of architectures called neocognitrons by Fukushima and afterwards the convolutional neural networks by Jan LeCun, who also implemented them on efficient hardware digital signal processors. And basically what is special about these architectures is localized and shared weights that endow the architecture with translation invariance. So if I want to classify an image of a cat as a cat, I don't care where the cat is located in the image. And initially, the computer vision community snobbed these methods, but everything changed about 12 years ago when the compute power allowed to develop and train sufficiently large architectures that outperform significantly everything else that was based on handcrafted features, and the entire field has changed. These methods have created multi-billion industry. So deep learning was awarded the Turing Prize in 2019. That's the highest recognition in computer science, and the rest, as they say, is history. Now, if you come to problems in biology, so a typical example in virtual drug screening, for example, I give you a molecule and I want to predict its properties. So here I can still input a molecule in some vectorized form into a neural network. The problem is that I don't have usually a canonical order of the atoms that comprise this molecule, right? So this is, if you're asking, this is a molecule of caffeine, so we're all familiar with it. So here the problem that we need to deal with is what we call permutation invariance. So the properties of the molecules do not depend on how I order its atoms, right? Because it's just a representation for the computer. The molecule exists on its own. It doesn't know that we somehow ordered its atoms. And it also exists in three-dimensional space, right? So it's mathematically speaking, a geometric graph. So if I rotate or translate it, the properties will also not change. So here we are interested in what is called SE3 invariance, right? SE stands for the special Euclidean group. And it means that the properties of the molecule do not change if I position it differently in the space. So I hope you get the point. So this is a class of techniques that we call geometric deep learning. So it's a way how our data interacts with certain geometric transformations. So we subject our data to certain transformations, and we want the neural network architecture to respond correctly to these transformations. And probably the most prominent example of these ideas was in AlphaFold 2, a famous Deepmind protein folding program that predicted this three-dimensional structure of proteins. And it also had its own breakthrough, the kind of ImageNet moment in structural biology, famous CASP competition, where a newcomer, a team that actually, well, at least before, never worked on proteins, and outperformed everything that existed before. And well, I probably wouldn't say that the rest is history. Just yesterday, they were awarded the Nobel Prize in chemistry, but this is history that is being written now, because I think it's just the beginning of this field. Now, if we look at these two breakthroughs, so ImageNet, the breakthrough in computer vision, and breakthrough in protein structure prediction, so there is exactly a gap of 10 years between them. And there is this maxim in machine learning that in machine learning, data is more important than algorithms. And this was a trend that observed, I think, by Alex Wisner-Gross initially, that if you take the amount of time that passes from the introduction of an algorithm that makes the breakthrough versus the time that passes from the introduction of data set on which it was trained, actually it's much shorter time from the introduction of the data set. That was the case with ImageNet, for example. So the convolutional architecture was known from the late 80s. But the breakthrough happened in 2012. The dataset ImageNet was introduced in 2010. It's interesting that this trend is reversed with AlphaFold. The dataset existed for many years. So the protein database, for example, started already in the 70s. But the algorithms on which AlphaFold was trained, which were used in the AlphaFold, were relatively new. More or less starting with transformers in 2017 and equivariant architectures that became popular around the same time. So it points to a different, another important consideration, which is a correct choice of the problem. And if you look at the amount of data also on which AlphaFold was trained, it is tiny by machine learning standards. It might be large for biology, but it's very small. It's order of magnitude less than 10 years before that, the ImageNet data set that contained millions of images. AlphaFold was trained on slightly more than 100,000 structures. And this is a drop in the ocean compared to big systems like ChatGPT, which nowadays modern systems, I think, are trained on trillions of data points. So basically, it's a question of what makes machine learning successful in a particular problem, and I'm talking here about biological problems. So I wish I knew for sure, but we can probably find some common patterns. And these are roughly four following patterns. It's the availability of large, diverse data sets with very clear performance criterion. It's the internal structure of the data, and some form of degenerate solution space. So the data set, I think it goes without saying. So in case of the breakthrough in 2012, it was ImageNet. It was established in 2010. It had about 1,000 fine-grained categories of objects and more than 1 million manually annotated images. And the performance was evaluated as average test accuracy on this data set. So for proteins, it was CASP. It was a competition established in the 90s on PDB, on the protein database structure that was established in the 70s. And again, the performance criterion was very clear, so that was geometric similarity of the predicted and the ground truth structure. Now, the degenerate solution space, if we look at images, they are not just random points in high-dimensional space. Actually, if we visualize them as points in high-dimensional space, we see that they form some kind of low-dimensional structure. This, mathematically, this can be approximated as a manifold, and this is often referred to as manifold hypothesis. And there are many reasons why this is the case. Images tend to be self-similar. So I can take a part of a cat, for example, an ear, and find its copy, maybe some slightly transformed at another location. So there is similarity across spatial positions and different transformations and different scales. When it comes to proteins, so typically when people want to impress you that work on this field, they will say that the hypothetical number of proteins that might exist in the universe is something like 10 to the power 300. So this is a number that is impossible to comprehend, right? So the number of atoms in the universe is just 10 to the power 80. So we have hundreds of orders of magnitude here. So it's bigger than anything that you can imagine. It's bigger than the number of cats and dogs in the world. It's bigger than the US national debt. It's bigger, well, anything else. But the question is whether these proteins really exist in nature, and the answer is no. And the reason is that proteins result from evolution, and nature tends to reuse existing solutions. And probably a good illustration is this structure that is called the Tim-Burrell fold. So it's very ancient, it's very old. It existed probably for more than 3 billion years. And it comprises these little blocks that are called beta-alpha subblocks. It appears across evolution in something like 10% of all enzymes that we know, across functions and across species. So you can find it in humans, in bacteria, even in tomatoes. So they do different things. So these are enzymes that do different chemical reactions, but basically they all have these kind of structures, but they're completely unrelated to each other. So the last thing, the internal structure, this is probably well illustrated with text. So if you look at how systems like ChatGPT are trained, so they trained on what is called masked token prediction. So you take a sentence, you remove a word from the sentence, and you try to predict it from the context. Like in the first sentence, if I masked out the word bank, you will probably be able to predict it from the fact that it talks about something related to a river, right? And in the second case, it talks about finance, so bank is probably it comes in the sense of financial institution. So this is called, our ability to do it comes from what is called distributional hypothesis that tells that words that occur in the same context tend to have similar meanings. This also happens to be the case with proteins and the concept of multi-sequence alignment when we take evolutionary related species where proteins have evolved by mutation, but somehow if you align them, you see similar patterns, similar amino acid sequences, and similar three-dimensional structures. They also tend to have the same function. And because AlphaFold is trained on functional proteins that actually survive the evolution and do something useful, there is very strong signal to learn this distributional hypothesis. So basically, all this shows that AlphaFold is probably one of the most famous illustrations of machine learning working in biological problems. And we can ask the question, does it really give us a theory of protein folding, right? So do we discover some new laws of physics? Do we learn some new laws of physics by training this system on biological data? So the answer is probably no. So there is evidence that it doesn't. And my argument is that maybe it doesn't need to. So it doesn't really need to generalize. You can think of it as a kind of very complex, and I'm of course exaggerating and don't want to make bombastic claims, but basically it does pattern matching in a very sophisticated way and pieces together different structures and different sequences. And there is this maybe a little bit radical claim that now with machine learning and with AI systems we are entering a completely new age of scientific exploration, and it appeared famously as post-theory science in this Guardian article, that so much data had accumulated and computers are already so much better than us in finding relationship between them that soon the old scientific method of hypothesize, predict, and test would be relegated to the dustbin of history. Now, whether you subscribe to this theory or not, we are already seeing some efforts in building artificial intelligence scientists. It is interesting to think how science will look like, especially experimental life science will look like in the age where increasingly more of the hypothesis generation and scientific discovery is delegated to the machine. And in this context, we tried to introduce a concept that we call black box data, and probably protein folding is a good example here as well. So if you think of biological research in general, it was always done by scientists for human scientists, right? So protein folding initial models were based on our best understanding, basically physics-driven models of how proteins fold, and the data on which they were applied was data collected by biologists for biologists. So the move to black box software, right? So to machine learning that was done by Deepmind and AlphaFold, replaced the white box software with black box software. But it was still trained on white box data. So the next logical step, in my view, would be to replace the data sources that were originally built for humans by data sources that are not necessarily understandable by humans, but make only sense in conjunction with appropriate machine learning algorithms. This is what we call black box data. We can use it already in, for example, design of new molecules. So this is a startup I'm involved in called Vant AI, where we are building molecular glues. So these are new type of drugs that make proteins that normally would not interact with each other. And this is a data source that is significantly cheaper, up to 6 orders of magnitude, than standard structural biology methods such as X-ray crystallography and cryo-EM. So I would like to finish with maybe on a poetical note. This is a quote from Goethe. So as you probably know, he was not only a poet but also a scientist, and he studied with Spielmann, who was one of the last alchemists who believed in encheresis naturae, the so-called hand of nature, some kind of semi-magical force that breathed life into living things. And he famously put this mocking phrase in the mouth of Mephistopheles in the famous scene from Faust, that encheresis naturae is the chemical name by which dancers themselves unwittingly shame. And I think now relying overly optimistically on AI to solve biology in some way is a return to alchemy, or maybe this time to encheresis machine, at the hand of the machine. I believe that we must develop experimental technology, basically wet lab technology, together with AI methods, and not just think that AI will solve all the problems. So I will stop here and will be happy to answer any questions. Thank you.
All right, Michael. So in the interest of time, we will only permit one question from the audience, and the answer will have to be around 30 seconds because we are out of time. Any one question, you please may raise your hand. We'll give you the mic. I'll give you my mic. See, convenient for you. There you are.
Hi. Thanks for the presentation.
I will. Thank you.
Thanks for the presentation and the ideas shared here. I have maybe a two-part question. First, here you mentioned that the amount of possible proteins was not really in a way feasible, and that there are these laws that are followed. How was that integrated into the system of AlphaFold itself? In the sense, was it like the loss function or the architecture of it? And the second part is, if we can incorporate such ideas in like real life simulations, do you see it feasible to actually like simulate everything in the future?
Well, I'm not sure I can answer this in 30 seconds. So basically, one of the ways that this assumption comes is, first of all, in the form of distribution, right? So you learn in some way the distribution, but also in the form of multiple sequence alignment. Because multiple sequence alignment sends you a very strong signal, right? So it means that in organisms that actually survive the evolution, you have certain structures and certain sequences. So not everything is possible. Like not a random order of words in a sentence would not make any sense, right? You will not see these sentences in books, for example. So whether it will be possible to simulate everything, right, any physical system. So I'm a big believer, well, and this is yet to be proven, of course, that generative AI models can replace iterative solvers that are traditionally have been used in computational science. It will be a different way of thinking because you rely on something that is physically, mathematically correct, but maybe solves the wrong problem, because this is our best understanding of reality that probably loses and abstracts out a lot of different details. So if we give it up and we have a black box that generates something, and sometimes this something is garbage, but we have a way to check whether it's garbage, then we will just generate more solutions until we find the right one. I don't know if there are other questions.
No more questions. Big round of applause once again for Michael, please. Thank you.